Good afternoon, good evening. Um, Happy to have you with us today. Let me see. I think we're good. To start, it's 11 a.m. here in North Carolina. And we have the pleasure today of having Dr. Gayle Volk from the USDA ARS. Until very recently, she was the interim lead for the National Plant Germplasm System. Gayle is a plant physiology that focuses on improving the management of vegetatively propagated genetic resources at the USDA plant germplasm system. And she is using apple as a model to develop techniques that enable not only the preservation, but also the characterization and future use of genetic resources in apple. And she's the coordinator of an effort to build and release public training materials related to the conservation and use of plant genetic resources to make that work known by the world. I always say that if our government wants to charge me more taxes that will go to maintain or sustain the USDA germplasm collection, I will gladly do it. Not for other things, but just for that. Okay. So Gayle, it's a pleasure to have you with us. I'm going to stop sharing here, and I'm going to give you the floor to you. Thank you so much.
Great. Thank you for this opportunity. And now I'm going to share.
Yes. It's coming. Okay.
Okay. Let's see. I have a lot of things showing up. Okay. Well, thanks for this opportunity today. I look forward to sharing some of the work that I've done over the past 25 years that I've been working with the USDA National Plant Germplasm System. And I'm trying to emphasize some things where we've taken an idea and made something happen with it and taken it more from just a research paper or a publication to actually something that's out there in the community and really making a difference within the plant gene banking world. Let's see. Oh, hold on, I'm having a hard time.
Yeah, probably click the screen and then you'll probably be good to go. Yeah.
Okay, let's see. There we go. Okay, so I'm also going to give you a tiny bit about my background, just because I didn't plan on ending up in the career that I have, and I feel like I landed the most wonderful, happy accident in the world because I have loved this job since I started it. So I'm originally from Corvallis, Oregon, out in the state of Oregon, and I did my bachelor's degree at Colorado State University here in Colorado. I'm right on the campus now. And got a degree in biochemistry. Then I went over to Purdue University and decided I really wanted to focus on plants. So I got a master's degree in horticulture. And then I wanted to feed the world. So I went to Cornell University and got a PhD in plant physiology with minors in molecular biology and cell biology. And then I thought cell biology is really fun and microscopy is great. So I did a postdoc at Washington State University in cell biology. And then lo and behold, ended up at a position here at the Gene Bank in Colorado in a building and a program related to where I worked as an undergrad. So I was hired originally to develop methods to cryopreserve clonally propagated crops in the National Plant Germplasm System. And that's the Gene Bank for the US. And they hired me with no experience in gene banking and no experience in cryopreservation. So I was coming into the job as a newbie, and that actually ended up maybe working in my favor because I had a fresh look on things. And I wasn't trained by the traditional experts in the field, but I was instead coming in to this career with some skills. And they wanted me to apply my skills to this problem. So I came in with skills in horticulture, in plant tissue culture, cell biology, molecular biology, and gene expression. And I also came into it pregnant with my first child, whether I interviewed as well as started the job. And then I was excited to apply my skills and find solutions to a really interesting subject. So I'm not a plant breeder. And I'm honored to be a speaker and to be invited to be speaking in this seminar series because I have so much respect for plant breeders. I conserve the plant genetic resources that are the building blocks for those breeding programs. So I think the work that we're doing in gene banking is absolutely linked to that which the breeders are doing. We're just one step upstream, and we're hopefully the source of the materials that let you accomplish your goals. So plant gene banks are mostly seed banks. So most of them consist of large seed collections, and those seeds can most often be placed into cold storage, such as freezer conditions, for long-term preservation of large collections. This doesn't work for all seeds, but most gene banks kind of started as a seed bank system. And this works for many of our vegetables and grains and oil seeds that are key for human food and nutrition. We have over 1,750 plant gene banks around the world. And the storage of seeds is really economical. It helps us keep them for decades to centuries in safe storage. We can keep them very secure. We can duplicate them at multiple places to make sure they're safe. But they do occasionally require regeneration to make sure that their viability remains high. And in the seed banking process, that regeneration step is the most vulnerable because that's when the seeds have to go out into the field, and that's when bad things can happen. And it's also very labor intensive. But sometimes we can't store seeds to capture the targets that we're looking for. So in that case, we're working with clonal collections, and that's what I focus my work on. So I work on collections of plants that aren't propagated by seeds. Here in these photos, you can see garlic, you can see citrus, you can see date palm, potato cultivars. And in all these cases, and maybe about 75 more crops that fall into this category that are pretty major ones, seeds don't capture the genetic composition. And these crops are propagated instead by grafting or by bulbs or by cuttings or some other method that we call vegetative or clonal propagation. Sometimes they don't have seeds. Think of a banana. Or sometimes the seeds can't be stored. And many of our tropical crops fall into that category. In other cases, even if we could store the seeds, it could take many, many years for that seed to grow into something that a breeding program or a research program would actually find useful. Because it doesn't make a lot of sense to receive a seed and wait 10 years for it to reach reproductive maturity so you can use that resulting plant in a cross. It's way easier to work with something that's already in a mature state. This is a map of the U.S. National Plant Germplasm System, and it's overlaid on top of the USDA hardiness zone, or the climate map for the U.S., where it shows different regions for planting materials. And you can see that we're a really distributed system. Our gene bank covers over 20 different sites, and each of these sites are specialized on the crops that tend to grow well in those places, because we need to put them out in the field to regenerate the seeds, or we are keeping these field collections of these clonal crops. So we need to put them in a place where those fields of plants can grow. I'm located right there in the center in Fort Collins, Colorado, at our base backup collection. This is a map of the NPGS accessions that we have in our gene bank. We've got over 16,000 species, representing nearly 620,000 unique accessions of materials. Of those, about 575,000 accessions are maintained as seeds. So 93%, or a large percentage of our collection, is seed propagated. And then we have about 44,000 that are these clonally propagated collections. It's only 7%, but 44,000 plants maintained in tissue culture and in pots in greenhouses and in fields is a lot of material that we need to maintain. These are just some photos from across the system. On the left-hand side, up in the corner, you can see the regeneration plots of our alfalfa. They're in cages, so we can control the pollination and make sure we don't have any outcrossing that happens when those plants are regenerated in the field. Down on the bottom, we see soybeans being regenerated, and they don't have that problem. They're selfed, so we don't have to worry about them cross-pollinating with the other accessions in the field. Then these collections are maintained for the seeds in cold storage vaults across the country. We often have an active collection that may be at 4 degrees, which is the distribution material. And then there is ideally also a freezer storage or a -18-degree storage for the seeds that are maintained for the long term at that location. And then those sites serve as the primary distribution sites for these seeds. And then all of the locations should ideally be backing their material up here in Fort Collins, where we have the backup or base collection. And you can see our freezer vault up in the upper right-hand side with these large movable shelves and over 450,000 accessions in there. And then in the lower right, you can see our cryo vault. So we have over 50 huge cryo tanks full of plant and animal and microbial materials that are secured here in our building. Our collection also does our very best to characterize and evaluate materials as we have the capacity to do so. This is particularly important for wild collections or wild species and unadapted materials where we don't know much about this material that we have. And we want to make it useful to the breeding community. So the more data we can provide, the better. Here we have some small grain collections being regenerated where they collect data in Aberdeen, Idaho. You can see diversity of soybeans down in the lower left, some soybean seed viability assessments in the middle, our apple collection in the middle, upper picture, and then regenerating peas in the greenhouse in Pullman, Washington. So I mentioned distributions. And we are the largest distributor of plant genetic resources in the world. The USDA system sends out over 200,000 items each year, and we distribute them free of charge for research and for breeding and for education. So this isn't intended to be a free source of seeds for home gardeners or for personal use, but it is that we are here for breeding and for research, because we want our materials to be used and to have that impact and to justify why we maintain this very large collection. To find out information about our collection, you can go to our website. We use the GRIN Global Database. And you can go to the website shown in the lower right and do queries based on taxonomy or common name and find out about our collections and what we currently have available. You can also find out about the passport information and any evaluation and characterization data that we have. We're always adding more information to this database, so it's always in flux. Then after you identify materials, if there's something you need for your research, you can actually put it in a shopping cart and place an order. And then the scientists at the locations around the NPGS receive those orders and they process them and they ship them out. Now, within the US, it's a fairly straightforward process for most seeds. They can usually just go in the mail or by UPS. But for international, it gets more complicated, and that's because we have to abide by international regulations. So we need to work with the requester to have them obtain an import permit for their country. And then with that import permit, that will let the seeds legally enter into the country. We also need to understand the phytosanitary requirements of the receiving country. And if we need a phytosanitary certificate, we can provide what we have available, but we can't promise the seeds are necessarily free of some of the contaminants that a country might require. And that may not let us ship the material, but we'll do our very best. And ideally, we're working with we have to be working with the requester to sort all these things out before the seeds go in the mail. And then the new thing that we added this year is accepting the SMTA. This is the Standard Material Transfer Agreement because we must comply with the International Treaty on Plant Genetic Resources for Food and Agriculture. And we comply by including an SMTA acceptance as part of all of our international shipments. Okay, now I'm going to tell you a little bit about what we do in Fort Collins. So, as I mentioned, we're the base collection. We have about 82% of the collection of the materials of our gene bank backed up as seeds in our cold vault room. You can see right here. We also have about 15% of the clonal collections secured, and those go into liquid nitrogen. We store them as either dormant buds or shoot tips or pollen, or sometimes seeds, especially for something like an intermediate or a recalcitrant type seed. Now, my research works on these clonal crops. So we have all these collections, such as the citrus in the field. We have strawberries in the greenhouse. There's garlic in cold storage. We have in vitro collections. And then we have these cryo storage collections. So we're trying to back up all these really vulnerable collections in liquid nitrogen. And we want to do this especially for these clonal crops because they're in places where hurricanes or disasters or diseases can happen, and we can lose the materials. And sometimes that happens, and we don't want that to happen. So we develop methods and we cryopreserve these collections and put them safe in liquid nitrogen as a backup. Now, another strategy we use only when we have to is making a duplicate collection at another location. But this is really expensive because these field collections cost a lot to maintain. So we try our best to put things into liquid nitrogen, and then they're safe for a very long time with only the cost of liquid nitrogen required. And that's what I was hired to do, was to develop these methods to cryopreserve materials in liquid nitrogen. And now I've grown my job to many, many, many more responsibilities over the past 25 years. So this is just showing the vast array of types of things we work on. And this is one reason why working in gene banks and in plant genetic diversity and conservation is so much fun, is because we get to work on a wide variety of crops. So I've worked with citrus and with grape and with kiwi fruit and with pear and garlic and others. And we have developed methods to cryopreserve our collections. And then we've actually shifted it from just research into implementation. So we're actually using these methods to back up the materials and make them safe. And it's not as easy as it sounds. So developing a method was one thing. That was writing the paper. That was identifying how to cryopreserve things. But scaling it up to cryopreserve hundreds of materials and keep track of them and all the necessary documentation is quite a big deal. And we learned that by going through the process. I'm just going to talk about a couple of these cryopreservation methods that we regularly use. And the first one I'll talk about is apple. And for apple, we cryopreserve dormant buds. This is a method that was developed long before I got in our building, about a decade before I arrived. And it was developed in about 1990 by other scientists here. And we continue to use this method today. We can also cryopreserve shoot tips for materials that are not hardy, like dormant buds of apple. And so we can take them either from plants in the greenhouse or screenhouse, such as for garlic or citrus, or we can take them from tissue culture plants. And for that, we use grapes and potatoes and kiwi fruits and strawberries and others. This is just one slide about cryopreserving dormant buds. We harvest them in January at a very cold location in the upper northeast part of the US. We dry them to about 30% moisture content, package them, cool them to -30, plunge them into liquid nitrogen, and keep them in the vapor phase of liquid nitrogen at -185 degrees. And we've got between 2,400 and 2,500 apple accessions cryopreserved using this method. When we want to pull them out, we can take out that dormant bud and we can graft it onto a rootstock and grow a new plant. So it's a way that we've been able to keep materials for over 35 years now in our cryotanks and pull them out when we need to replace materials in the field that are lost. So it's about 2,500 accessions we have right now that have been cryopreserved here in Fort Collins between 1988 and 2024. Another, this method we developed, this was a challenge for us. We wanted to develop a method to cryopreserve citrus because there's some major diseases in citrus called Huang Lung Bing, or citrus greening, and it's impacting the citrus industry in the US very significantly. Now, this used to be just in Florida, and now the disease has spread to the citrus production areas in California. And our challenge is that our citrus collection is also in California, and it is surrounded by some fields that have trees that have the infection. So the collection has been safeguarded by putting it into a screen house, but still, it's really vulnerable. In fact, a couple years ago, there was a storm and we got a tear in the greenhouse. And so we had to frantically fix that tear and retest all the materials in the screenhouse to make sure that the HLB disease is not in our collection. Thankfully, we were fine. So we were asked to develop a method to cryopreserve citrus about 15 years ago. And we found a method that doesn't require us to put the collection into tissue culture. Up on the left-hand side here, you can see that we can take these twigs of materials of the fresh flush of growth from the citrus, send it to Fort Collins, we surface sterilize it, we excise out 1 millimeter shoot tips, treat it with cryoprotectants, load it onto foils, like little pieces of aluminum foil. These are like 1 millimeter, they're tiny. And then we plunge it into liquid nitrogen and put them into a vial and put them into and store them. Then when it's time to recover them, for some plants, we can plate them onto media, but in this particular case, we have to micrograft them. So we take that little tiny shoot tip, micrograft it onto our rootstock in tissue culture, and you can see the lower right-hand plant of a citrus that's recovering after cryostorage. These are just pictures showing that process where we're harvesting the twigs, surface sterilizing them in the second picture, excising the bud in the third picture, and then treating it with cryoprotectants in little petri dishes. This is including things like glycerol and sucrose and DMSO and ethylene glycol. And this helps suck the water out so that when we cryopreserve it, it doesn't have freezable water. Because if we had water, that would cause ice and that would kill them. So then they get plunged into liquid nitrogen and stored. When it's time to pull them out, we can take them out of liquid nitrogen, put them onto that little rootstock, the etiolated plant, make a little ledge, trim off the bottom of the shoot tip, place it on there, and then watch it grow. So 2 weeks later, we have a little plant starting to grow. 4 weeks, we have a rooted plant. 8 weeks, we're almost getting too large to continue to grow in the test tube, and we can move it out to the greenhouse. They are flowering within about a year, and that's really exciting because that means we didn't go back to the juvenile state and we stayed mature. So this is a method that we developed in my lab, and we've now cryopreserved over 500 accessions of citrus to protect the sanitized part of our national collection at our building. So this is an example of research going to implementation and actually securing a collection. And to my knowledge, it's the only cryopreserved, clonally propagated citrus collection in the world. Okay, now I'm going to give an example out of apple. And in addition to doing cryopreservation, I use apple as a model to understand diversity in our national gene bank for a clonal collection. We have over 3,000 trees maintained in Geneva, New York, that maintains everything from representative wild species to crabapple ornamentals, to hard cider varieties, to heritage varieties, to modern varieties of apple. And you can see a range in the diversity of the apple fruit on the right-hand side. Here's another picture that I took 2 years ago in the Geneva apple collection, showing the wide range of trees we have in the field, as well as the fruit that I collected from those trees. Oops. So what we did, and we started this work about 20 years ago, is we used first SSR markers, and now we use a 20K SNP array to understand the diversity in the collection. And we're using this in collaboration with other countries around the world who've also genotyped their collection for apple using the same array. And now we can compare the collections. It's really hard for us to share material because of quarantine restrictions, but it's easy for us to share data. So we can better understand if our materials have the right names for cultivars on them, as well as if we have the right species names, or if we have hybridization issues within the materials and they aren't labeled correctly. This helps us better maintain our collections, and it gives us a reference set to identify the cultivars for some unknowns. So there's a lot of tools we can deliver to our customers and stakeholders about diversity in our collection, as well as the accuracy of the materials in our collection using genotypic data. One thing we did is we took this genotypic data for our collection, and we developed a collaboration with Washington State University. And they developed a website where you can go to myfruittree.org and find and send in a leaf of an unknown apple tree, and they'll tell you the identity of it by matching it with the materials in our National Gene Bank, or by matching it with the genotypes of other samples that have already been submitted. And this has been really popular. We have national parks with old heritage apple trees that are maybe 100 or 150 years old around the country, and they don't know what we have in these old trees, and they're dying. So now they have a way that they can send these in for either small batches or large batches of leaves, and find out what the identities are and whether this is something that they need to invest resources in to conserve because it's rare and unusual, or if it's really common and you can just replace it by ordering another tree from a nursery. So these projects that I've worked on in both citrus and apple tend to go big. And this is what I really like about my job, is that you get opportunities to have an impact that's more than just local. And in this case, I co-authored 2 different strategies. One is the global strategy to conserve apple genetic resources, and another is to conserve the citrus genetic resources of the world. So we did a big survey to understand what kind of collection materials are out there and what their vulnerabilities are, and worked with the community to develop some proposed ideas about how we can all work together to save these important crops on a global basis. Okay, this next section I'm going to talk about is another project that I've plunged into. And about maybe 6 years ago, I was asked to develop a training program for gene banks because the USDA didn't have anything like this, and we didn't have access to training material that was really relevant for our own use within the system. There's a lot of folks that were retiring in the gene bank, and we wanted to share or save that knowledge and information and then share it. So I was asked to develop a program, and I did. We wrote a grant. We first we surveyed to figure out what the needs were. Then we wrote a grant, and in that grant, between Colorado State University, USDA, and Iowa State University, we built a program called GRIN-U. So you can go to grin-u.org website. It's all free, and you can browse over 200 items. You can also sort for them and query them based on topics of interest. They are ebook chapters and ebooks. They are videos. They are infographics in multiple languages, and other relevant content that sometimes somebody else has developed, and we decided that it's so good that we're going to link to it on our database as well. If you, so please feel free to browse this site and learn about what we've shared. We continue to post new information. And to find out about that, you can go to the upper right-hand corner and subscribe. And then you'll get an email, or then you'll have an opportunity to get an email every quarter, so not very often, to say the new things that we've posted in the last quarter. So you don't have to constantly be searching the database to see if there's something that you missed. We also built this so that anyone at gene banks around the world, or even in universities, could use this for training materials. So we wanted it to be a source where a professor might need a video or some content about seed storage or about cryopreservation or about breeding and pre-breeding, and they could come in here and grab a video that we've already prepared. We have an associated YouTube channel. It's Grin U Education. And we currently have over 108 videos posted on this YouTube channel, where these are mostly in-house videos about our collections, or stories about evolution or domestication or diversity that also could be relevant to breeding programs or to gene bank programs who are looking for some information about something that perhaps the average YouTube creator doesn't talk about. If you're interested in this, you can also subscribe to our YouTube channel. And this is available both within the US and internationally. We write ebooks. So we have a number of ebooks, these are just some of them, where we've been creating content and putting it out for the world to use on a number of different topics. These are all a work in progress. Some of them are considered finished, but some of them we add more chapters when we have time. And this is an advantage of working with an ebook platform, is that we can go in and add more material when we have the opportunity to create it. And it's not like a hard book where you create one edition and it gets old. In this case, we have a more living, active supply of content. This is just one of the ebooks we released a couple of years ago, and this is on conserving and using climate-ready plant collections. So this is talking about the effects of climate change, both on gene banks, as well as how plants within gene banks can be used in plant breeding programs for adaptation and mitigation. Pat Byrne was my collaborator, or actually Tara Moreau and Pat Byrne were collaborators in developing this book. And Pat is the one who wrote the section on plant breeding. There's a lot of content there. So this is, again, useful for the breeding community to learn about that connection between gene bank resources and outcomes. Another book that I have a lot of passion for is success stories. So we decided that that we really needed to tell our story better. The gene banks sometimes get taken for granted because we're just the supplier of the raw materials. And we needed to better document how critical we are for both American and international agriculture. So I have an ongoing effort to collect success stories. And if anyone has any, I would love to learn about them. I'm doing them mostly for USDA, but I'm also have some international stories as well. And what we're doing is documenting materials and successes to show that these materials have been critical to overcome problems. There's a one-page, these are all chapters in an ebook. And again, we keep adding to it. There's over 20 in there right now. One of those chapters is about the hazelnut gassaway cultivar, which gave Eastern fire blight resistance, Eastern filbert blight resistance to the modern cultivars that are grown that massively allowed for the expansion of the industry for growing hazelnuts or filberts in the United States. Here's a couple more examples. We have a sweet potato called Tinian that has Fusarium wilt resistance. And this sweet potato, let's see, This original variety was obtained in 1946 from the island of Tinian in the Northern Mariana Islands, and it's been maintained in our National Gene Bank. This material was used in breeding programs to incorporate the resistance to Fusarium wilt. And currently, all the US commercial lines that are grown have this resistance. So Fusarium wilt is not a major concern in the industry anymore. And that industry is a $676 million industry. If we look at budgets, this one particular accession gives more than tenfold return on investment in a single year. So the gene bank costs about $50 million to be run in the US. And here we are getting a $676 million industry impact every year. Here's another example, and this one's for blueberry. This is a blueberry cultivar called Florida 4D that has allowed us to grow new varieties of blueberries in the southern part of the US. So this particular cultivar or accession in our gene bank was a native plant that was grown in a pasture in Florida, which is a really warm state, a southern state in the US, and was found in the 1950s. And this material was used to cross with northern commercial blueberries that are more commonly known varieties. And they developed a southern blueberry. So now this new blueberry type went from having the original cultivar requiring 1,000 hours of chilling a year to now needing 0 hours of chilling. So we can grow blueberries in warm climates. So this has totally expanded the industry to both subtropical and tropical regions for growing blueberries within the US and internationally. Okay, we also have infographics on our website. So these are, we have 4 sets of them right now. The one on the left here shows how plant genetic resources are the key to global food security. So it provides some connections between why we should have gene banks and their use in breeding programs. And the one on the right here talks about main functions of a gene bank. I didn't really cover this today, but gene banks have functions of acquisition and of maintaining, evaluating and characterizing, regenerating, documenting, backing it up, and distributing. And so this infographic shares that kind of information. And thanks to some colleagues of mine, we've made these available in a number of different languages. So you can have this content in English, French, Spanish, Arabic, Chinese, and Portuguese. These are free posters that you can download from GreenU, and you can either print them on a 1-page piece of paper, or they could be printed as poster size. So they're very sizable and friendly to use, and you're welcome to use them. Then we have 2 more posters here, and these again can be shown as large posters or as 1-page handouts. We have the one on the left-hand side for about the National Plant Germplasm System in the US, available in English and Spanish, and it gives more details about our gene bank, as well as the one on the right-hand side that talks about the partnership opportunities between gene banks and botanic gardens, because there's no way we can do everything, so we should be partnering together, and we have complementary efforts. Okay, I wanted just to spend a couple minutes talking about an opportunity I had this year. And I was invited to serve as the acting national program leader for our National Gene Bank for about 10 months this year. And I did it remotely from Colorado, where I'm based. But it was a fabulous opportunity to learn more about administering a gene bank, both in terms of policy development and standardization of procedures, and then coordination among all of our different locations at both a national and international level. I've also learned that this is a huge position and a huge job. And I decided that I'm really happy in my day job. So I didn't apply for the final job. We've recently hired a new scientist into this position. Her name is Dr. Neha Kathari. But I also, since it's such a big job, I didn't have the opportunity to do everything because I was still doing my day job. So I picked some aspects of it where I thought I could have some particular impact. And one of those was better understanding U.S. obligations for the International Plant Treaty and how we can be compliant and engage more fully with our necessary requirements on that. And this involved a lot of the SMTA work and incorporating that into our database. I've also spent a lot of time doing team building and visiting the different locations within our gene bank. And while I was there, I brought my phone and I captured a lot of video. So we will soon be releasing these videos to our GreenU YouTube and website. And it's fun to share information about what I learned at these locations and have our curators and our technicians talk about what they do and about and show the passion that they have for maintaining these vast collections. I'm also really excited about these impact and success stories. There are so many stories out there, and this is a huge opportunity for engagement with the plant breeding community because it's often the plant breeders that have the great stories. So I've learned that if I ask our gene bank people, they may not know their impacts. But if I ask a breeding community, everyone in the room can talk about accessions that they've used that have made a difference for their programs. And those are the stories that I'd like to document and share. I'm also excited to share more about the NPGS. I've done a lot of outreach, a lot of conferences, and a lot of engagement activities to better, to not let us be a secret and to really get the word out that we're available both as a resource of information as well as a resource of plant materials for the appropriate purposes. And then the final thing I've spent time on is, and I've started and I'm going to continue, is to really work on building these partnerships. Because there's no way that we can do all the work that needs to be done in plant genetic resource conservation in the US alone. So we need to be partnering with the botanic gardens, with native land managers, with small collections, with maybe local collections or universities to document what each other has and to figure out how we can complement each other and make sure that these materials remain secure and safe. And all that while still doing my day job. Okay, so I'm going to wrap up and just acknowledge that, of course, none of these things are done alone. I have lots of collaborators. I've splashed some of their photos up here on the screen from various projects over the years, and I've got so many more that I did not specifically mention today. And with that, I'm going to conclude, and I'm wondering if there are any questions.
Thank you so much, Gail. What a wonderful presentation. And yeah, I do have a lot of questions, and I can start with one while other people post their questions online.
Okay.
I did post the link to the Green U on the YouTube channel, so people has it.
Okay.
I know that this is not the most sexy topic for politicians to finance, and you are living that as a lot of other people that preserves genetic resources in the world are living that, okay? And I wonder if you can tell us a bit what we can do, what you're doing to raise awareness. I mean, all the stories are probably part of that, yeah.
Sure. Yeah, so I didn't go into that today, but I would love to talk about that subject also because I've spent a lot of time sharing that information in all my presentations this year. The US government, actually Congress, asked us, our gene bank, to write a strategic plan in 2018. And this is to address the backlogs of what we have in our system and where we're falling behind. Like, how many regenerations do we have to do to get caught up? How many materials aren't cryopreserved? How much data do we need to collect to phenotype and characterize our collections? And this was a real soul-searching exercise for us because we developed there are a number of people. Our former program lead, Dr. Peter Bretting, was responsible for this, but lots of us helped him gather the information needed. and write the documents. There's actually 4 documents, and those documents are hundreds of pages long. One of them is a 2-page infographic. And we've changed the way, this is something I did this year, as I started talking a little bit differently. We used to say, look at how much amazing material we have in our gene bank. Look at how many distributions we do. Look at how large we are. I've been trying to change the conversation to say, yes, we have this amazing resource. It's critical for global security, global food security. And it's vulnerable. And we've admitted we've made these documents all public. So we admit that we have backlogs. We admit we can't do it all ourselves. We can't do it alone. And we actually need more funding to do so. We're also engaging actively with stakeholder communities who have some ability to impact funding in their ways.
Well, I
So
hope those documents are not cryopreserved.
well, the everything's up on the web. All of these documents are totally available. And what
in Congress,
oh,
I
yeah.
mean that they don't put it in the freezer. Yeah.
I know, exactly. But the one thing I'm also doing, I quickly realized that our documents, we have this massive hundreds of pages of material that nobody's going to read. So I'm actually going through and making crop by crop summaries of that content so that we can share 1-pagers to customers and stakeholders and let them know the situation for the crops that they care about. It's getting engagement, that's for sure. We've spent the year talking about it, and now we're getting action and traction.
So we have one question in the chat, and I encourage participants to post other questions. It's about getting like vegetatively propagated accessions internationally. Can they move? Participant from Ethiopia is interested in that particular accession of sweet potato, but what is the process? Because with seeds is one process, but I imagine with in vitro is another level of complexity, maybe.
It is.
Okay.
Generally speaking, materials in tissue culture are easier to share internationally than materials that are from the field. Every country has their own quarantine and recipient requirements. So this needs to be I would recommend that you put in a request to the gene bank into GRIN, our database, and start a conversation. So if you put in an order, then the gene bank will reach out to you and they will tell you what they need. And they'll tell you perhaps what material that you like, the documentation that we would need before we could send it. And hopefully there might be a way. The US has ways to receive material from other countries, and they go through a quarantine process. So they get checked or cleaned before they go into the public, but we have a way to receive things. And ideally, we're working, other countries have those same processes, but there's no promises. We can do the best.
And also, you are part of Genesis, isn't it?
USDA is in Genesis, yes. You can also put your order in Genesis also. And any order placed on USDA materials in Genesis comes to us. We just get an email from Genesis saying, hey, you have an order. And then what you do is you get an email saying, would you please put the order into GRIN, and we'll take care of the order our usual way. So, but yeah, you can browse Genesis to get information too.
Okay, it's a question coming from Majuma. What are the most effective molecular techniques for assessing genetic diversity variation in tree species, and how can researchers secure funding for projects related to forest conservation and biodiversity through genetic analysis?
Whoa, let's see. In terms of the best methods, I think they are, I think you find out where the technology is for the specific crop that you're looking in. If it is a SNP system, if it's a sequencing type system, the SSRs are still valuable, but I would never develop them specifically now. But if they're available and they're already established within the community, then that is also a system that could be applicable. But usually it's larger genomic technologies that people are using. Funding is a challenge sometimes. I think you really need to couch that proposal in the economic importance of understanding this diversity and conservation. Maybe there are opportunities for agroforestry connections, or maybe there are opportunities for indigenous agricultural methods, or wild utilized plants type systems. And perhaps that could help increase the possibility of funding opportunities.
Good. Although the scope that you show is amazing, but I would say that doesn't represent much of the actual diversity in the world for different crops. I mean, some crops have been kind of very thoroughly collected, but others. And my question is, has there been any effort put into preservation of pollen, for example, as an easy way to, I know you don't present the identity of the particular genotype, but at least you present the genetic load of that genotype by preserving pollen, which is a small amount under cryopreservation. Has there been any progress on that?
Yeah, I took out that slide. I do that
Oh,
also.
okay.
But I didn't coordinate that question with you. So,
Oh,
yeah,
no, we
we
didn't.
developed a method for cryopreserving pollen. I currently every spring we cryopreserve pollen from some of our crops that we don't have other methods to preserve them that are clonally propagated. An example is date palm. We have, I think, about 42 male date palm trees in our national collection. They make a lot of pollen. It's easy to cryopreserve their pollen. We have them all backed up in liquid nitrogen now as pollen. I continue to work on the things that make catkins. So the hazelnuts, the walnuts, the pecans. We cryopreserve pollen from those every year because we can get a lot of pollen very, very easily. We've done some prunus pollen as well. Sure, there are opportunities for lots more of this, but again, I don't see it as the primary thrust of our efforts. So we have a few hundred of them in cryo right now. And we do it every spring for a month or two, with one person processing any of the material that people can send us. The other challenge with pollen is that we are completely dependent upon the active site providing us with that pollen. They can't ship us a tree and have us collect the pollen. So they need to do all the collection. They need to keep it all separate. They need to do the initial drying for like overnight, and then the sifting. And then we receive it and we adjust the moisture content and put it into liquid nitrogen. And then we do the viability assessment by in vitro germination. But yeah, it's
That's
a
good.
complementary process. All of these,
Yeah.
we got to dry everything to make this work.
Right. Comment, a question from Matthew Blair. It says that the posters seems to be a good resource, and he suggests that what about printing them in giant size and posting them in airports somewhere that people can read it, okay,
Oh, I
and be
love
aware.
that.
Yeah.
They're free.
It's
You can do it.
how are you promoting your extension type of work? That's his question.
Oh, great question.
Mhm.
Oh, well, I include those slides about GreenU and the YouTube channel and every single presentation I give. I also encourage our program leads to do this. And I think it's catching on. The idea is, how can we get this information out there? And I love showing up at various places and seeing our posters on the wall. In fact, I was just at the U.S. Crop Science Society, or the CSSA meetings, the Crop Science Society of America meetings this week. And I was talking to a book author, and I was telling him about our content. And I said, oh, you're a popular book author. You're on the speaking circuit. Maybe you can share information about the gene bank as part of your discussions of your books. And he was really interested. So I'm attempting one by one to help maybe get more people to help share the word, because there's no way we can do it alone.
Okay.
It's growing. Sure.
Jen Yu from Taiwan is asking that he would like to further understand the apple accessions they have collected, and if would it be possible to extract DNA and use the MyFruit tree system for analysis, or would there be a better method to do that?
Have him contact me. We can talk. There
Okay.
are other ways available for, I don't know if we're talking about cultivars of apple or if we're talking about wild species of apple. The myfruittree.org, that's for cultivar names. And
Oh, that's for
it
culture.
matches
Okay.
you up to US cultivar names. we have other systems for species identification. So I'm not quite
Oh, I
sure,
see.
and the ornamentals fall into a slightly different category. We don't have necessarily a good genotyping method to identify the ornamentals. There is so much diversity in apple that the SNP arrays that are built for the breeding program that we're using for the genotyping are really cultivar and the closest wild relative bases. So we use
Jen,
those
you,
for the
sorry, I have posted Gail's email address there for you to contact her, please.
Okay, sure.
Well, wow. Thank you so much, Gail. I'm gonna close the meeting. I'm gonna share. Okay. See. Well, thank you so much for the participation of everybody. Thank you, Gail, for an amazing presentation. Um, Next week, we'll have the last seminar of the series by Dr. Julianne Swankert from a private company in the Netherlands that is working in promoting F1 hybrid potato seed for commercial use. A very interesting and novel approach. I will encourage people to attend this seminar because I think it's going to be very, very informative and novel, okay? Until then, you guys have a great weekend, and I look forward to seeing you next week. Thank you so much, Gail,
Oh, thank
for all
you.
you do and for the presentation, okay?
Thank you so much.
All right. Bye-bye.
Bye-bye.